Welcome everyone to our annual barbecue and awards ceremony. Of course, without the barbecue and without the camaraderie and face-to-face contact. I hope you found some time to enjoy the Ferdinand's ice cream nonetheless. It's today to present these awards to just a small subset of our very deserving faculty and staff. So before I do that, I want to go through a number of thank yous, as you can imagine. So first, I want to thank Dr. Bob Maley. He can't be with us today, but I wanted to thank him for all he did for the college as interim dean. His willingness to step up was really appreciated. I heard it from many people. Even before I arrived, Bob was communicative, transparent, and engaged. He was eager to support you all and to welcome me into my new role. He's settling into his new role as Veterinary Clinical Science Chair, and I very much look forward to our ongoing work together. So thank you, Bob. I wish you were here. He'd be right here. I'd like now to introduce a man who actually needs no introduction, your former dean and interim provost, Dr. Brian Slinker. Brian agreed to attend these awards ceremonies in great Cougar spirit, as always, to acknowledge and honor the faculty and staff recipients of these awards. Of course, the barbecue would have been even better, but these are very strange times. So Dr. Slinker served as your dean for 10 years. He was a champion for faculty, staff, and students. He oversaw the building of 2 premier research facilities, the Paul G. Allen Center for Global Animal Health and the Veterinary Biomedical Research building that houses integrative physiology and neuroscience. He also helped to secure funding for the new Washington Diagnostic Lab, Waddle, and he built a strong regional WEMU program and was passionate about innovation and education. He has not left us. He and his wife, Kathy, continue to be proud Pullman residents and very strong supporters of the college. So Brian, before you speak, I'd like to present you with your retirement certificate. As we were unable to do this before, it looks like this. You can come over and grab it, but we can touch in plastic. There you go. Do you have this, Henry? All right, and with that, I will give the stage to Brian.
Thank you, Dori, and thanks for having me back. It's been a weird year for all of us. It was almost a year ago that I suddenly departed as dean and became interim provost, September 30th, 2019 to be exact. And I never really got to properly say goodbye, so I really appreciate being able to be here and celebrate with you these staff awards. I've done many things over my life, and the dean's job had a lot of cool stuff, but the highlight of my year was always our barbecue and the opportunity to recognize our staff. So I'm really appreciative of being able to be here and do that today. As Dori said, Kathy and I are in Pullman, and in normal non-pandemic times, we'd be around and we'd get to see you. Someday that'll happen again, and so we're staying here. And I just want to thank you all, not only for the work you did this last year, Bob, the whole leadership team, all of you for how you had to step up, but also thank you for all these nearly 3 decades of me on the faculty. You've been great friends and colleagues, and the college is stronger for you and will continue to be strong. The provost job was one I never wanted. And my 10 months doing that convinced me that that was the correct judgment. But we have a really good new provost that I really think is going to shine and work with you all to move the university ahead. And the absolute coolest thing, though, about being provost was I got to hire my own successor as dean. And so, Dori, welcome. It's been an honor to be your dean. And like I said, I'm around, and with Dori, I know you're in good hands. We're in good hands. And so thank you all.
All right, thank you, Brian. And I do very much trust that he and Kathy will be around. I've spent some time with both of them, and they're so passionate about the college, so he will be here. I also want to thank all the people who made this award ceremony possible. Julie Watkins, Executive Assistant to the Dean. She's a consummate professional, very caring, and has worked so hard to try to make this perfect for us all. I want to thank the members of the Dean's Staff Advisory Council, That's led by Laura Kinslow, and this is the committee who picks the awards for the staff awards. And also members of the research committee who select our faculty research awards, and that is chaired by Dr. Lee Nodler. So before we start, I also want to make one comment about the tremendous commitment of our essential faculty and staff at Waddell and the Veterinary Teaching Hospital. They've been working through COVID since the very beginning, and these are truly challenging times. So essential staff have had to balance the concerns about personal health, family and friends, work demands, their care for their community, and their responsibility with a lot of unknowns. Inevitable delays in communication and policy that quite simply arise from this having been the very first pandemic that we've had to deal with, and hopefully the last. But our patients are still there. Animals are needed now more than ever for that critical human-animal bond, and testing still happens, as you know, at Waddell. So I really thank you. It's been such a pleasure trying to work through this with people who are as committed as our critical and essential staff and faculty. We appreciate you. So we're going to start today with the Length of Service Awards. So employee recognition is a vital part of the Washington State University. We recognize the contributions of our employees, and they're critical to fulfilling the university mission. So the following Washington State University College of Veterinary members are being recognized for 5 years of service. Philip Vaughn, Jessica Hoffman, Laura Lockhard, Kevin Repp, Billy Weston, Heather Beckner, Glenn Duncan, Bridie Gillespie, Crystal Quigley, Savannah Tiedemann Mueller, Lane Wallet, Tony Mertz, Da-Chang Mo, Joy Winatanyan, Troy Jackson, Trang Volkarakis, Travis Burke, Casey Burton, Kelsey Dunmeyer, Marcia Ginther, Jen Gold, Cynthia Kiepert, Martin Alejandro Suarez, Cynthia Zering, Paige Grossman, Robert Burke Demonin, John Davis, Ryan McLaughlin, Claudia Moore, and Stephen Roberts. The following WSU College of Veterinary Medicine community members are now being recognized for 10 years of service. Sarah Pepper, Andy Allen, Jacob Lowry, Sally Madsen Botters, Danielle Nelson, Jennifer Babcock, Cheryl Williams, and James Peters. The following community members are being recognized for 15 years of service. They include Blake Baker, Terry Hassold, Pat Hunt, Bill Durnell, Denise Hotchkiss, Lisa McEwen, Matt Mikas, Gary Halderson, Victoria Haluby, Devendra Shah, and Catherine Noble. And the following members are being recognized for 20 years of service. They include Bonnie Campbell, Lynn Nelson, Bob Maley, Lon Smith, Rick DeBose, Lynn Haley, Heiko Jansen, Leslie Sprunger, Jennifer Watts, Brooke Wolf, and Doug Call. We're going to skip now to members who've been recognized for 30 years of service. Those include John Gay, Patricia Duffy, Charlie Powell, Henry Moore, Tim Basler. We also have 2 people in our community who are being recognized for 35 years of service, and they include Tracy Topping and David De Avila. Members who've been recognized for 40 years of service is kind of like that game. It keeps on getting higher. Include Patty Haley, Michael Smurden. And we have one faculty member in our community who is now being recognized for 50 years of service, and that's Dr. David Preer. So congratulations to all. Thank you so much for your service. It's truly a pleasure having faculty and staff who are so committed to long-term loyalty to the college and to the Washington State University. So in this, I also want to specifically recognize our Quarter Century Club inductees. So the Quarter Century Club, which I'm now going to call QCC, is Washington State University's longest running employee recognition program. It has been active since 1934. Each year, the Quarter Century Club recognizes faculty and staff who have reached the impressive milestone of 25 years of service to the university. Historically, I'm going to say this now, they host an annual breakfast to honor staff and faculty, and as you can imagine what comes next, this fall this has been canceled. But as this breakfast was originally canceled, they're going to honor their inductees and a dedication and experience nonetheless. So to recognize new QCC members in 2020, this year's 57 inductees from across the state will be individually featured on the web page later this year. Honorees will also receive a letter of congratulations from President Kirk Schultz and a framed QCC certificate and a commemorative QCC challenge coin. The QCC comprises more than 2,300 individuals who have long formed the backbone of this university. So our inductees this year from the College of Veterinary Medicine include Lynette Kinzer, Terry Olson, and Kathleen McMenamin Snekvik. All right, so we're going to move now to the Outstanding Individual and Team Staff Awards. These award recipients are nominated for their extraordinary work and are the employees that people simply cannot live without. I want to truly also thank the nominators. These are busy people who take the time to publicly acknowledge and highlight the work of those around them and to truly acknowledge the staff, and this is a really great honor. So none of this, none of all of the college could happen without our staff. The award consists of a certificate and a check. And so as I call you up, go ahead and come down and you'll have those awards and then you'll have your photo taken. So the first recipient is Kristin Roche. So Kristin is a scientific assistant in Dr. Dana Shaw's lab in veterinary microbiology and pathology. Kristin's being recognized for her attention to detail, her initiative, her high standards, motivation for herself and others. She's a clear communicator and is highly collegial with others within and outside of her lab. With COVID-19, she methodically closed down the lab to protect all the valuable resources. It was greatly appreciated. She's described as resourceful, outstanding, and a pleasure to work with. Congratulations, Kristin. And there you go. So the second recipient is Caitlin Estalia. Caitlin is the lead program coordinator, student services area, and was nominated by her colleagues and supervisors. So Caitlin directly or indirectly supports the education of about 530 of our professional students. She's consistently efficient, creative, and effective in her work, always personable and professional interactions with colleagues. During COVID, Caitlin worked with college leadership to make extensive adjustments to student clinical schedules in ways that minimized the impact on their education and allowed for our students to graduate with their clinical year intact. She then shifted, after doing that, to reworking 135 student schedules who were then entering clinics. Caitlin again rose to the challenge and worked tirelessly to essentially reorganize the entire year. She's described as compassionate, hardworking, highly knowledgeable, and proactive, among many other things. So congratulations, Caitlin. Yeah, exactly. All right. The third recipient is Jane Hummel. So Jane is a licensed equine veterinary technician who is nominated by the entire equine team, faculty, residents, and an intern. Jane has outstanding horse handling skills and puts her patients first. She teaches the students with patience and kindness and is consistently thinking ahead of what needs to be done for the day. Jane is an upbeat team player, flexible, highly communicative, and always does her job with a smile, which of course we can rarely see now. She has single-handedly organized our equipment room. That was very appreciated. I would have appreciated that too. She has outstanding leadership qualities, and not only is she an incredible asset to the team, but she's one of the finest technicians with whom we've worked. Jane is truly appreciated for her work by all. She's also earned a clinical teaching award from the students. Congratulations, Jane. Yeah, exactly. The fourth recipient, is Kristina Karn. Kristina, otherwise Nina, I understand, is a licensed veterinary technician in small animal surgery. Nina is the go-to person for students, staff, and faculty for anything small animal surgery. She does tremendous work under pressure while remaining calm and professional. She's an excellent organizer, knowledgeable, above and beyond her training level, and she has a knack for anticipating patients' needs, which makes surgery safer for pets. She's taken ownership of her position and her duties in a commendable way. Her supervisor described her as one of the best surgery technicians she's ever worked with. Nina has a strong work ethic and holds herself and others to high standards. She personifies initiative, creativity, and perceptiveness, and as further testimony to her outgoing class of 2019, awarded her as the outstanding staff member. So congratulations, Nina. All right, our fifth recipient is Casey Burton. Casey is the veterinary technician in oncology. Casey is being recognized by her supervisor for being an eager, cheerful attitude, for great work skills, and a love of patience. Casey has amazing flexibility, and she's a true team player. She's had to manage significant shifts in technical support and caseload during COVID. You oversee radiation and medical oncology, and you handle tremendous variation in daily tasks, which is really appreciated. She's the go-to person for catheterization. I love that. I needed that a lot in my world. It's a great skill. She fills in, she steps up, and she works well with house officers and students. She has a genuine interest in everything done in oncology, and apparently is a true lifelong learner who's eager to learn more. So thank you, Casey. Congratulations. Thank you. Mm-hmm. Thanks, you're welcome. All right, we're now going to move to 2 staff team awards. These awards normally come with lunch with the dean, which is a most wonderful acknowledgement, if I must say so myself. Somehow I get to benefit from their hard work, so that seems really reasonable. And we are going to make this happen in a slightly different way sometime this year. So the first team award, do I have these? So the first team award goes to the small animal surgery technician team. This team consists of Rachel Kimbrell, Kristina Karn, Christina Ojunen, Chris Dumas, and Christian Bowe. This team runs 3 services within one: soft tissue, orthopedics, and emergent. And they can only do so because they work so well together and communicate. Under the calm and steady leadership of Rachel, they make sure 5 operating rooms are running smoothly. They turn around rooms in an amazingly short time frame and yet meet the highest standards for cleanliness. So it was bragged that you have perfect surveillance cultures. They're spoken of in the highest terms by their faculty, who show tremendous trust in their decisions and skills. They bring a level of knowledge and professionalism to the teaching hospital that allows for the surgical team to do some of the most complicated surgeries in the country in veterinary medicine. They worked together to handle the addition of an extra surgery service this last year, the emergent service, and they help other services in the hospital. With COVID, they had to shift their schedules significantly to stretch and cover, especially when there were no students present. They think outside the box and they're solution focused. Congratulations, thank you all. Please turn around and have your pitches taken in that direction. Or, I think, okay, they know what they're doing. You're correct. Do you want to come down? You can come down the opposite way. We'll allow it. Congratulations, you guys. That's all right. Congratulations.
Thank you very much.
Congratulations
Thank
again. Congratulations. Congratulations. Thank you. All right. The second team award goes to the Veterinary Clinical Pharmacology Laboratory, or the MDR1 testing lab. This team includes Becky Connors, Judy Arneson, Kelly Brown, and Neil Burke. This team is recognized. I'll let them filter in here for a moment. We're not used to being in person these days. Yeah, who invented those stairs? All right, hello. So this team is recognized for consistently bringing their A game to the college. These 4 individuals must perform at the highest level of efficiency and accuracy and professionalism on a daily basis. Their success in doing so has made the canine MDR1 genotyping test the most critical veterinary genetic test. The multiple integrated steps that this team takes on a daily basis to run an accurate and efficient customer-based service is truly appreciated. They enhance the reputation of the college as the veterinary school with expertise in individualized medicine. And that's true. I'd heard about you long before I came. The team is also generous with their time when it comes to helping other college members, and they're innovative, they're problem solvers, they're energetic, committed, and dedicated to the pets we serve, their owners and veterinarians, and they're very loyal to the college. So congratulations to you all, and thank you for your service. Yay. Thank you, Dr. Neely. Yep, you can come down here if you'd like, or you can go right. Oh, you have to turn around for your picture. Sorry. Just turn around for your picture. Right up here. Yep. If you'd like to come down and elbow bump, you can. You do not have to. What's that? Okay, perfect. Yeah, I know, exactly. Either way, go ahead and go back up. Sure, that's perfect.
Thank you.
You are so welcome. Thanks for joining us.
Thank you for doing this.
Absolutely. Yeah, I know. All right. We're going to move now to our last 2 awards. These are our Faculty Research Awards. Each year, our research faculty nominees are ranked and selected by the College Research Committee. The first award is the Zoetis Award for Veterinary Research Excellence. This award is sponsored by Zoetis and is given to faculty who have made significant contributions to research that benefits veterinary medicine. Animals suffer from many of the diseases in which our college faculty are actively engaged in research, including viral and bacterial infections, cancer, obesity, heart disease, and reproductive problems, among others. This award is presented with a plaque and a cash award of $1,000 from Zoetis. This year, the Zoetis Award for Veterinary Research Excellence goes to Dr. Santanu Bose from Veterinary Microbiology and Pathology. So Dr. Bose runs a renowned and well-respected research program that focuses on innate immunity against viruses, virus-based cancer therapeutics, and during the pandemic, he stepped up to do coronavirus. He uses cellular models and mouse models, and he studies the mechanisms regulating inflammation. Dr. Bo's research is very relevant to veterinary medicine. Important diseases with significant inflammatory components include pneumonia, cancer, diabetes, and arthritis, all of which negatively impact cats, dogs, horses, and cattle. His work has tremendous potential in development of anti-inflammatory therapeutics to combat inflammatory diseases not only in humans, but also in veterinary patients. For example, he recently received a US patent for development of S100A9 based therapeutics to combat pneumonia. This novel anti-inflammatory therapeutic could be extended to treat pneumonia in animals. Dr. Bose has been recognized as one of the Washington State University's top industry-engaged faculty. He also received the Washington Research Foundation's Innovative Technology Gift Award. He was selected as the Caroline Engel Distinguished Professor in Research on Infectious Diseases in 2015 in recognition of his substantial, impactful contributions to research. Several of his works have published and featured in the Faculty of 1000 and in journals like Nature Reviews Immunology. He consistently secures extramural funding, and he has 4 US patents and licensed inventions. He loves to teach and mentor. He's served on the graduate committees of many graduate students, and he mentors postdoctoral fellows, undergraduates, and high school students. He's highly collegial, he's unselfish, and he always has the best interests of the department and the college in mind. So congratulations, Dr. Bose. It's truly a pleasure. You've earned this. You can go ahead and come down, and the Zoetis plaque and check are in the mail. We're a little bit early for our ceremony this year, but at least come down. You can have your picture taken. You can do an elbow bump if you'd like with your dean and with my. Congratulations, Ms. Santano.
Thank you.
Best of luck. It's just like it's coming. It's in the mail. Yeah, exactly. So it has a great plaque. It's coming. All right. So now we're going to take a few minutes, and Dr. Bose has a nice recorded talk about his research.
Thank you very much for listening to my talk. And I'd like to thank everybody who nominated me and selected me for the Zoetis Award this year. And I'm greatly honored to be this year's Zoetis Award recipient. We primarily study with 2 respiratory viruses, influenza A virus or flu, and respiratory syncytial virus or RSV. And more recently, we have started working with human coronaviruses, including the COVID-19 causing SARS-CoV-2. As you all know, that influenza virus can cause pandemic. And one of the major pandemic caused by influenza virus or flu was the 1918 Spanish flu, which killed about 20 to 40 million people. So coronaviruses can also cause pandemic, as we are going through right now. The COVID-19 pandemic, and so far almost 1 million people are dead due to this current COVID-19 crisis. So what are some of the hallmarks of respiratory virus infection? One of the major thing that happens with these viruses is that they infect the respiratory tract, basically the lungs and the airway of the. The other thing that happens with these viruses is that they can cause severe pneumonia, which is severe inflammation of the lung that can cause death. what is inflammation? So inflammation is part of viral innate immunity. And inflammation is required for a cellular defense against viruses. So when a virus like flu or coronavirus infects our lungs, it launches a regulated pro-inflammatory response by triggering production of cytokines like TNF and IL-6, which then forms a regulated inflammation, which brings in the adaptive immune response comprising of T cells and B cells that can clear the virus from the lungs. So inflammation is good for us when it is regulated, and it is important for host defense and also to maintain a homeostasis. But the problem is that if the inflammation is dysregulated, if there is an excessive inflammation in the lung, that can trigger severe pneumonia. And that's what happens with the deaths associated with flu and coronaviruses like SARS-CoV-2. What are the problems with the zoonotic respiratory viruses like flu and coronaviruses? One of the major problems with these viruses is that they are prone to frequent mutations. Therefore, the existing antiviral therapeutic or the vaccine will not against these mutated strains. The strains can also jump from animals and birds to humans, causing deadly disease. And once again, the vaccine of the antiviral therapeutics will not work against these new strains. And it is hard to predict where, what, and when a deadly strain may arise that can cause a pandemic. And the most important thing to really recognize is the fact that it takes more than a year to develop and distribute vaccine for the pandemic strain. And we know of this crisis that we are going through right now because we still don't have a vaccine for SARS-CoV-2 that is causing the COVID-19 pandemic. So what are the solutions? So one of the solutions is that there is a need for us to be prepared for the pandemic. And the way to be prepared for the pandemic is to develop a broad spectrum anti-inflammatory, anti-pneumonia therapeutic agent that targets cellular factors. And the reason that you need to target the cellular factors is because it's independent of the viral factors which are prone to mutations. We immediately need broad spectrum anti-pneumonia therapeutic to control the spread of the pandemic virus strain, and so that in the meantime, a vaccine is developed and distributed to public. So you need that broad spectrum therapeutic to control the mortality, widespread mortality among the public. So the goal is to develop a broad spectrum host-directed therapeutic anti-inflammatory agents that can be given to patients to control pneumonia. And that will result in a far better survival outcome for patients infected with flu or with SARS-CoV-2. Can we target a host-derived factor to control respiratory virus-associated pneumonia? Yes, we can. So for example, I'm just going to talk about this protein called S100A9 protein that we identified as one of the host factors that can regulate pneumonia. So from now on, I'm going to call S100A9 protein as A9. So A9 protein was detected in patients infected with flu, and it was detected in critically ill flu-infected patients. And so we looked at whether flu can induce expression and production of A9. So we use a mice model where we infected the mice with flu via intranasally or intratracheally, so the virus can go to the lungs. And as you can see here, that indeed flu can induce production of A9 as shown here. And also when we took the lung slices and we did an immunohistochemistry, we can see expression of flu in the lungs as shown here as the red rings. So next, we wanted to see the role of A9 in causing inflammation in the lung. So we used a control IgG antibody and S100A9 blocking antibody. So this antibody blocks the function of A9. And we took the lungs from flu-infected and uninfected mice, and we stained with H&E. As you can see in the mock uninfected mice, the airways are nice and open, and it looks very clean, so the lungs look very healthy. After flu infection, as you can see here, there's a full-blown pneumonia because of the cellular infiltrates and the cellular debris that come to the airway and clogs it up. But interestingly, what we found is that if we use an A9 antibody, the inflammation is drastically reduced and the disease is controlled. As you can see, the airway is not clogged up. In terms of survivability of the flu-infected mice, we saw that A9 can protect flu-infected mice from mortality. When we use the IgG control antibody, the flu-infected mice, all of them, died within 6 days. But when we use the A9 antibody, those mice, almost 80% of those mice can survive past 6 days, and you could see them extending up until. And more recently, the A9 protein was detected in COVID-19 patients, and we're surprised to see that this A9 was detected in critically ill COVID-19 patient infected with SARS-CoV-2. So we did some studies here in our lab to see if A9 is induced by coronavirus. So as you can see, when we infect mice with coronavirus and we treat the lungs, we can see expression of A9 in the lungs of the coronavirus infected mice. Also, when we infected macrophages with human coronavirus, we can see robust induction of A9 in the macrophages. So basically, what we know from our studies that A9 is a critical host factor contributing to inflammation and pneumonia during respiratory virus infection. And based on our data with flu and some of the data that is coming out with our studies with SARS-CoV-2, we propose a schematic model of how A9 may be involved in the disease process. So when the respiratory viruses like flu and CoV-2 infect cells, it produces A9, which goes to the neighboring cells and turn on a signaling pathway, resulting in production of inflammatory cytokines and chemokines, which then gets out of the cells, causing airway inflammation, pneumonia, and death. But if we can block A9's action on the cells by using a blocking antibody, we can prevent production of inflammatory cytokines and chemokines so that there is less airway inflammation, there is less pneumonia, and there is reduced mortality. So the whole purpose of our project was to develop A9-based broad-spectrum anti-inflammatory, anti-pneumonia therapeutics to combat pneumonia associated with any strain of flu and coronavirus so that there is cross-protective effect of agents against these viruses. Our next goal is to advance this technology to clinics so that the antibody could be given to patients infected with these viruses. I'd like to thank everybody for listening to my talk. Thank you.
So welcome back. Thank you again, Santanu, and congratulations. So our second and final faculty award is the Dean's Outstanding Junior Faculty Research Award. This award's sponsored by the Dean and designed to recognize an outstanding junior faculty member who's already established in themselves as innovators and leaders in their chosen fields. So the college faculty who are pre-tenure or clinical eligible faculty within 6 years of starting their research careers are eligible. The award is presented with a framed certificate of award. So this year, our award goes to Dr. Winu Talyanan. I'm going to call you Joy, if you're okay with that. She joined the School of Molecular Biosciences at Washington State as an assistant professor 5 years ago. She hit the ground running in both research and teaching. She received an R1 grant in 2019 on her main project that involves the actions of steroids on oviduct function, and she received a diversity supplement on that award. She also investigates the use of protease inhibitor to inhibit semen liquefaction in the female reproductive tract. The observation is very novel, and if the compound is proven to be an effective contraceptive approach, the significance is extremely high. Since arriving at Washington State, she's published in high-impact reproductive biology journals, as well as excellent journals with a more general audience. Along with her research effort and productivity, she excels in teaching and mentoring. She works with both undergraduate and graduate students. Her director stated in his letter that her student reviews are among the most positive he has ever encountered. Several of her reviews ended with the comment, "I just love her," which I think is great. Similarly, she has volunteered for service within the unit and has performed in an outstanding manner. Joy is being recognized as one of the brightest and potentially new investigators in the field. She's junior, very energetic, with the drive and the intellect to be successful, and we are very lucky to have her. So congratulations. Come down for your elbow bump. Congratulations, you're welcome.
Thank you.
And do pick up your certificate. All right, and now we're going to hear a little bit about Joy's research.
Thank you very much for giving me an opportunity to share my research here with you today. In our lab, we're focusing on two main research projects. The first one is evaluating the action of steroid hormones in the oviduct. And the second one is the development of the new contraceptive method for women. So I'm going to talk about this first project. Is that why are we focusing on this oviduct? It's because this is the place where fertilization and pre-implantation embryo development take place. So you can see here, this is a fertilization, and these are all pre-implantation embryo that is developing inside the oviduct before they implant into the uterus, where they stay to the rest of their pregnancy. And our interest is that we ask the question, how does steroid hormones involve in these processes? And these female steroid hormones include estrogen and progesterone. So why estrogen and progesterone? We know that through the use of previous study from other studies, they show that female mice with the global deletion of both either estrogen receptor, which is encoded by ESR1 gene, or progesterone receptor, or PGR, the female are infertile. If you look at this gross anatomy of the female reproductive tract, you can see the vast difference between the uterus and the ovary of the oviduct of the knockout of ESR1, as well as the progesterone receptor, or PGR. So from these study years back, they show and they know that estrogen and progesterone are important for the function of the uterus and the ovaries. But what about the oviduct? We still virtually unknown what is the role of estrogen or progesterone in the oviductal function. So we ask, what are the action of these steroid hormones in the oviduct, right? And we use a conditional knockout mouse model where we ablated estrogen receptor or progesterone receptor only in the epithelial cells of the oviduct. And we collect the embryo at day 3.5. From the female, and this is what you would find in the control mice, that there's a blastocyst, and these are morula stage embryo. And in our epithelial deletion of estrogen receptor, you can see that these are all dead embryo. We never see a viable embryo in this conditional knockout oviduct at all. And for the deletion of progesterone receptor in the epithelial cell, we also found a lot of the dead embryo within the oviduct, and some embryo that didn't develop normally. And right now we're trying to find what is the cause of these embryo developmental defects and how does estrogen and progesterone are involved in these processes. Next, we evaluate or develop a new contraceptive method for women. And we use or target the semen liquefaction process and to be used as a novel female control contraceptive method. So normally the semen is ejaculated to the anterior wall of the vagina, and within this semen there's a protein called seminal gel, which is the seminal gelin 1 and 2. And the enzyme from the prostate gland called prostate-specific antigen, which are the serine protease, hydrolyze this seminal gel, and then the gels are degraded and the semen is liquefied, and the sperm can swim to the fertilization place, which is in the oviduct. And we target this by using the serine protease inhibitor to block the action of PSA enzyme. And we test this, the idea using human semen. And we found that with the fresh semen, if you incubate it at 4 degrees, which inactivate the enzyme, you can see those are the gel that is present. But when you incubate it at 37 degrees, so the enzyme becomes to work and the gel is hydrolyzed. So there's no more gel. And when we add protease inhibitor, these are the gel that we observed again. And we collect the sperm from this portion and we evaluate how many sperm that are swimming. This is what we found, that with the treatment of the protease inhibitor, more than 90% of the sperm are now immotile, which create a infertility-like environment for the sperm, because if you have more than 60% of the sperm that are immotile, then based on World Health Organization criterion, you are now infertile. So we're testing that this idea in the female mice, and also we aiming to go through using the female rhesus monkey to test this idea whether it works in the primates or not. So we're doing that with the collaborator. So those are the two main projects, and I would like to thank people that actually did the work, people in my lab, both current and the former lab members, those are here, and also the WSU faculty, my mentoring committee, Drs. Prue, Oatley, and Hunt, and other collaborators here at WSU, and other places across the US, and also the funding source, both from the NIH and the intramural funding from WSU. And lastly, I would like to thank the CVM Research Committee and also Dr. Borjesson that give me this award, as well as our director, Dr. Crispo, that nominating me for this award. I am honored to be a part of this strong research community within the CVM, and I'm happy to do research here many more years to come. Thank you very much.
All right, so congratulations again, Joy. It's wonderful, your achievements, really appreciated. And thank you all for joining us this afternoon, spending some time with us. Again, to Julie and staff who helped organize this, and Brian Slinger for making his appearance again with us. And next year we're just going to have to celebrate twice as much. So really appreciate all the faculty and staff. And when we're back in person, we're going to make up for all this missed time with lots of great events. So appreciate you being here. Have a great rest of afternoon, and we'll see you next year at the Staff Awards.